Safety Testing of Cosmetic Products. Overview of established methods and new approach methodologies, or NAMs.
In this presentation, our goal is to offer you a quick summary of the research and conclusions. Please follow the links to review the research in further details. Thanks to all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share, and subscribe.
Cosmetic products need to have a proven efficacy combined with a comprehensive toxicological assessment. Before the current cosmetic regulation, the 7th Amendment to the European Cosmetic Directive has banned animal testing for cosmetic products and cosmetic ingredients in 2004 and 2009 respectively.
An increasing number of alternatives to animal testing has been developed and validated for safety and efficacy testing of cosmetic products and cosmetic ingredients. For example, 2D cell culture models derived from human skin can be used to evaluate anti-inflammatory properties or to predict skin sensitization potential. 3D human skin equivalent models are used to evaluate skin irritation potential, and excised human skin is used as the gold standard for the evaluation of dermal absorption.
The aim of this manuscript is to give an overview of the main in vitro and ex vivo alternative models used in the safety testing of cosmetic products with a focus on regulatory requirements, genotoxicity potential, skin sensitization potential, skin and eye irritation endocrine properties, and dermal absorption.
In conclusion, a wide range of in vitro models for safety testing of cosmetic products and cosmetic ingredients has been developed and adopted in test guidelines.
There is still an increasing need, largely driven by regulatory authorities and industry, to develop in vitro models to predict carcinogenicity, repeat dose toxicity, and reproductive toxicity, for which no alternative in vitro methods are currently available.